Okay, good afternoon, everyone. Welcome to our February seminar. I'm very pleased today to introduce our seminar speaker, Foster Jacobs. Foster is a dedicated PhD student at the University of Minnesota School of Public Health Division of Environmental Health Sciences. And his academic journey is impressive. He has a bachelor degree in biochemistry and Spanish study from University of Minnesota, and he has continued his graduate studies here at the UMN with a focusing on environmental toxicology. He is a valuable member of the Balbo Lab. Foster's commitment to advancing our understanding of chemical carcinogenesis has taken him from the research labs at the Masonic Cancer Center to the International Agency for Research on Cancer in Lyon, France. And over the past 6 years, he has made significant contributions to our field by developing and applying mass spectrometry-based methods, resulting in multiple high-impact publications. His work has been also recognized through several awards and fellowships, and he is a recipient of, just to mention few, the Kelly Coleman Toxicology Fellowship and Chateaubriand Fellowship. On a personal note, it's especially gratifying to introduce Foster today because we are part of the same program at the School of Public Health, and he has been an exceptional colleague and friend. He's always ready to brainstorm, troubleshoot, and he always offers insightful perspectives during our hallway conversation. And his enthusiasm and collaborative spirit make him a very important part of our community. So I'm very pleased to welcome Foster, and Foster, take it away.
Great. Well, thank you, Alexandra, for that very kind introduction. I'm very happy to be here to give this talk. So let's get into it. So like I said, and like Alexandra said, I am a member of the Balbo Lab. And so today I'm going to be talking to you all about how our lab is harnessing the power of DNA adductomics to study DNA mutations. So there are several environmental risk factors that are associated with cancer development. However, many questions remain regarding these associations. For example, what are the molecular mechanisms explaining these associations? Or why is it that not all people exposed get cancer? So what we are trying to do is to understand and define the sequence of events that lead from exposure to cancer at the molecular level. And so all of these different exposures that I just showed you fit into the scheme for chemical carcinogenesis, where you can visualize how these exposures can lead to the formation of DNA adducts that eventually lead to mutations that can cause cancer. And so typically in our lab, we are focusing on identifying and characterizing DNA adducts. And so we analyze DNA adducts using mass spectrometry. And with our high resolution accurate mass instruments, we have been able to develop methods that allow us to simultaneously detect DNA adducts resulting from a variety of different exposures. And so in order to understand how we use mass spec to analyze DNA adducts, it's important to briefly go over the structural components of a DNA adduct after the DNA has been hydrolyzed and is ready for our analysis. What remains is the deoxyribose sugar, the nucleobase, and the actual adduct, right? The portion that is covalently bound to the DNA. And so we take advantage of these common features in order to detect the DNA adducts with mass spec, whether that's looking for the loss of the deoxyribose sugar or the loss of one of the 4 DNA bases. But it is important to note that these are not the only neutral losses we can look for. These are just some of the typical neutral losses for many different types of DNA adducts. And so when it comes to DNA adductomics, there are various approaches we can take. With the first being a targeted approach, where we can target a large number of known DNA adducts simultaneously. And we can also perform an untargeted approach, where we can simultaneously screen for both known and unknown DNA adducts in a single run on the mass spec. And additionally, our ability to analyze this untargeted data is improving thanks to recent software advancements as well. And the DNA adduct data that I will be showing you today will be from a targeted approach, where we targeted a list of adducts that had previously been identified using our untargeted screening approach in similar samples. So we know that smoking tobacco can cause lung cancer by inducing DNA damage. However, we really need to define what exactly is happening at the molecular level following an exposure, as this would shed light onto why some people are more susceptible to tobacco-mediated carcinogenesis than others. So this is where DNA adductomics come into play. And so, in order to define what is happening at the molecular level and to try and figure out which DNA addicts are playing a role in tobacco mediated lung cancer, we took a step back and developed an animal model utilizing NNK, a potent lung carcinogen in tobacco smoke that is known to induce DNA damage and lead to lung cancer. And in this model, NNK induces lung cancer after about 70 weeks of chronic exposure. And we've also co-exposed these animals to an inflammatory agent, lipopolysaccharide, which I will call LPS, because inflammation is a major risk factor for lung cancer and increases the severity of lung carcinogenesis in animal models exposed to NNK, where both the size and the numbers of the tumors are increased. And so we did this in order to mimic the effect of a smoker who has COPD and has inflammation in their lungs, as smokers with COPD are 4 to 6 times more likely to develop lung cancer than smokers without this condition. So in this project, we are aiming to better understand the role of DNA addicts in NNK-induced lung carcinogenesis, and we are investigating the effects of inflammation in intensifying this process in order to try and identify the addicts that are driving tobacco-mediated lung carcinogenesis. And so, to identify these DNA addicts, we utilized a time course to capture different periods within the pre oncogenic process before tumors can form. Right? The idea here is that we can identify the DNA addicts that eventually lead to mutations that can cause lung cancer. And if our hypothesis is correct, the addicts that are driving the carcinogenesis will be present at higher levels in the rats exposed to NNK plus LPS than those exposed to NNK only. And of course, we do have the appropriate controls here as well, where we have animals exposed to LPS only and animals that did not have a certain chemical exposure. And so, when working with our NNK animal model, it's important to understand the metabolism of NNK. Shown here is a very simplified version of the metabolism, but what's important to understand from this is that the metabolism of NNK is complex and leads to a variety of metabolites that can form different types of DNA addicts, including methylated addicts, pyridyl oxybutylation addicts, also known as POB addicts, and also other mono and cross-link addicts as well, due to the production of formaldehyde in the metabolism as well. So, in order to analyze several different types of DNA addicts in a single sample, our lab has developed an approach to compare samples for mass spec analysis, giving us the ability to characterize different classes of DNA addicts simultaneously. And typically, our analyses generate a lot of data. So we have a variety of workflows for interpreting our mass spec data, all depending on the goal of the project at hand. And so, in this experiment, we have looked at many different types of DNA addicts. But today, I'm primarily going to be showing some data from 3 different groups of addicts that showed some interesting results. With the first being a methylated DNA addict. So, the addict that I wanted to show you here is the extensively studied 0,6-methylguanine. We analyzed this addict using an established assay that does not require high resolution mass spectrometry and allows us to perform absolute quantitation using our internal standard. And the first thing that I'd like to point out here is that this adduct was not detected in our controls and the animals exposed to LPS only, suggesting that our model and approach are working as expected, as this adduct is not endogenous to this model. And for those who aren't as familiar with looking at this type of mass spec data, we expect that the analyte of interest, 0,6-methylguanine, shown in the top panel here, we expect it to colute at the same time as the labeled internal standard, which in this case is a deuterated form of 0,6-methylguanine seen in the bottom channel here, right? And so if you take a look at these chromatograms, you can see that there is a peak for 0,6-methylguanine at the same time as the internal standard for both NNK and NNK plus LPS, right? You can see that there's 2 peaks here at pretty much the same time. Same thing over here, right? That peak may not be as big as this one, but it's still a nice peak at the same time as the internal standard. And if we look at the control, there's no clear peak at the same time as the internal standard. And then if we look at the LPS only, again, there's not a clear peak at the same time as that internal standard. But that blob that you can see at the beginning here is just other junk that's in the sample that has a similar mass. But we can see it because we're not using high resolution mass spec in this scenario. But also, since that blob is not colluding with the internal standard, we know it's not our analyte of interest. And so to quantify the levels here in the case for the NNK and NNK plus LPS samples, what we do is we integrate these peaks to get the area, and then that data is normalized to the area of the internal standard. If anyone here has additional questions about how this works, feel free to reach out to me, or I'm sure maybe you have a colleague that may be familiar with analyzing mass spec data as well, as I can see many names of people I know and recognize in attendance today who would definitely be able to explain this as well. So moving forward here, I wanted to show you the levels of 0,6-methylguanine that we were able to detect in the rats exposed to NNK and NNK plus LPS. First, I'd like to point to what we observed in the NNK only samples, where there is a gradual increase over time as previously reported and thus, as we expected. However, unlike our prediction, the co-exposure to LPS did not increase the levels of this DNA addict when compared to the NNK only in any of the time points, as you can see here, where in each case, the co-exposure actually led to decreased levels, which we found to be interesting, despite not being in accordance with our hypothesis. So next, I will show you some data on the POB and PHB addicts. But before I dive into this, I wanted to make a note here that the rest of the DNA addict data that I will show was all generated using a single method where we targeted more than 20 different analytes. And this was done using one of the high resolution mass spectrometers that our lab has access to, the Orbitrap Lumos, for those who are familiar, which I would say is a great instrument for harnessing the power of DNA adductomics. So let's get into it. So the first addict that I wanted to talk to you about here is O2-PHB-DT. As you may remember from the metabolism of NNK, this addict is directly derived from NNK metabolism, and therefore it should not be detected in our samples that were not exposed to NNK. And as you can see here, that indeed is the case, where we can see very nicely that there are peaks for both the NNK and NNK plus LPS samples, but there's not for the control or in the LPS only. And two things you might have noticed here that are different from the 0,6-methylguanine slide is that I don't have an internal standard for this addict, and I'm also displaying these numbers over here. So these numbers indicate that the mass of the addict itself is 392.1821, also known as the parent ion. But the mass that I'm extracting from the data here is called the product ion, which is an ion that is a direct result from from fragmenting the parent ion in the mass spec. And in this case, this product ion for O2-PHBDT has a mass of 276.1347. Well, what does this correspond to, you may ask? Well, if we look at the structure of this DNA addict, you might be able to recognize a couple of the features I talked about earlier when I was describing how DNA adductomics work, particularly the deoxyribose sugar seen here. So when we fragment this molecule, we will observe that the deoxyribose sugar is one of the first features to fall off. And it's important to note that this loss of DR is typical for many types of DNA addicts. And that is because this bond right here, right, is between the rest of the molecule and the DR is one of the weakest bonds in this molecule. And so following the loss of DR, the resulting mass is 276.1347. And so that is what we are looking for when we analyze this data. 276.1347 that originates from and is the product ion of the parent ion with a mass of 392.1821. But sometimes it isn't enough just to know the loss of DR to confirm that the analyte you are looking at is indeed your analyte of interest, as the loss of DR may not be the primary product following fragmentation of the parent ion, and thus the levels may be too low for detection. So it's usually ideal to have multiple product ions that you can look for, and this is especially the case when you don't have a labeled internal standard, as is the case here. And so I've shown you how this addict can lose the deoxyribose sugar, But as you've probably gathered, that's not the only place this molecule can fragment. This is a well-reported adduct, and others have reported that this adduct can also fragment right here, resulting in the loss of the deoxyribose sugar and the thymine nucleobase, right? Which leaves us with the PHB here with a mass of 150.0913. And so for the mass spec people out there, I did just want to make a note that all this data was generated in positive ion mode. So all the masses I'm reporting here are all protonated masses. So back to O2-PHBDT, here again we see the loss of the deoxyribose sugar, and as we can see, the mass of the PHB colludes perfectly with the loss of DR, providing further evidence that this is O2-PHBDT, despite the fact that we don't have an internal standard to confirm with. And when we look at the mass spectrum for this analyte, on the far right, you'll notice just the remainder of the parent ion that wasn't fragmented, right, that 392.1814. And then you can see the loss of DR, and also we can see the PHB, right, indicating that the product ions we have looked at here are also the major fragments of O2-PHBDT that are produced when using our method. And so when looking at the levels of this adduct and those for the closely related O2-POBDT, we observed that again, for the NNK only, for both of these adducts, they increased over time as previously reported. However, this time, while the co-exposure did not enhance these adducts for all of the time points as predicted, there is an increase in adduct level that happens earlier with this co-exposure compared to the NNK only, as observed here in the 5-week time point in the case for both of these adducts. And so now I'd like to show some data for the various crosslinks that we are able to detect. But first off, it's important to note that these crosslinks are formed by formaldehyde, and formaldehyde is endogenous. So therefore, they are observed in our control samples. So, as you can see here, the previously reported DG-DG formaldehyde crosslink was detected in all of our samples. But, as you may notice, even though we're using high-resolution accurate mass spectrometry, and despite looking for the loss of the deoxyribose sugar from the parent mass of 547.2008, we still have 3, beautiful might I add, peaks in every exposure condition. And additionally, this is one of the many adducts that we don't have an internal standard for. So, the question is, how do we determine which peak is the DG-DG formaldehyde crosslink? So first, when it's a previously reported adduct, I like to draw the structure. And as I've talked about already, the first fragment that we should see would be the loss of that deoxyribose sugar, right, resulting in a mass of 431.1534, which is the product ion that I showed results in 3 different peaks. However, if we think about the features of this crosslink adduct, right, it has 2 DGs, and therefore it also has 2 DRs. So why not see if both DRs would fall off upon fragmentation in the mass spec? And so resulting in a product ion of 315.1060. And so here again, we see the first product ion, right, that loss of 1 DR. And here we have the loss of 2 DRs. And it turns out that this product ion perfectly colludes with the first of the 3 peaks, indicating that this first peak is likely our analyte of interest. And for further evidence, let's take a quick look at the mass spectrum for this analyte. So here we can see the product ions that we've already looked for, right? The loss of 1 and 2 DRs. However, there are other product ions in the spectra that are much more abundant. But it turns out these are product ions that are exactly what we would expect to see when it comes to a DG-DG formaldehyde crosslink, where we see DG, we see guanine, and we see a guanine with that formaldehyde linker still attached to it, right? And so these are all further pieces of evidence that corroborate the idea that this analyte is indeed the DG-DG formaldehyde crosslink, despite not having an internal standard for this. And so for all of these crosslinks that I'm about to show you data for, we really had to scrutinize the MS spectra like this in order to confirm their identities, as these are a relatively new class of adducts for us to be analyzing in mass like this. And so therefore, we don't have internal standards for most of these. And so when it comes to the levels of these DNA-DNA crosslinks, we observed here that for the NNK-only samples, there was an increase observed at the 1-week time points when compared to the controls, right? If we look right here and right here. But there wasn't really much difference from the endogenous levels of these addicts at the later time points, right? If we look right here. Yet, when we look at the co-exposure, the behavior is similar to the POB and PHB addicts when we compare to the NNK only, right? Where there are increases happening earlier for the NNK plus LPS in the earlier time points, right? At the like the 1 and 5 week time points in this case. So next, I'd like to talk about DNA RNA crosslinks, with the first being a DA guanosine formaldehyde crosslink, which happens to have an exact mass of 547.2008. So therefore, when I went to analyze this addict, looking for the loss of DR, I again found myself having to try and figure out which peak is my analyte of interest. Fortunately, however, I only had to look at the two peaks on the right, as I'd already determined that first one to be the DGDG formaldehyde crosslink. And so here we have the DA guanosine formaldehyde crosslink with DA from DNA and guanosine from RNA. And so just like the DG-DG crosslink, it loses DR, hence the three peaks. However, unlike that DG-DG crosslink, this analyte has a base from RNA. So therefore, instead of looking for the loss of a second deoxyribose, we can look for the loss of a ribose, right? Giving us a mass of 415.1585. And when we plug that in, we can see that the middle peak is our DNA RNA crosslink here in this case. And when looking at the spectrum for this crosslink addict, we see the loss of DR and R, of course, as we just showed. But it turns out that we can see pretty much every single product ion that we would expect to come from this analyte, which personally I found to be pretty cool, as this is not always the case when doing trace level analysis. And if you want to learn more about how we came across this DNA RNA crosslink using our untargeted screening approach, I would recommend checking out our group's paper on this, where we first detected these types of analytes in mice that were exposed to NNK. And so this is the paper. And if you read it, you'll also be able to learn about how highly motivated chemists can troubleshoot the synthesis and purification of a newly synthesized DNA addict standard. And this really was a big step for us, because now we can confirm when we see it, and we can develop quantitative assays for this crosslink using these new standards. So, we really appreciate all the work that went into synthesizing those standards. So thanks, Matt. And so when it comes to the levels of these DNA RNA crosslinks, the behavior is actually similar to the DNA DNA crosslinks, where we observed an increase in the NNK only when compared to the controls at the 1 week time point for most of these addicts, right? As you can see right here. But then in the co-exposure, we see again that there is a trend of increases happening earlier when compared to the NNK only, particularly the 1 week time point in this case. And so next, I want to briefly talk about one of the DNA protein crossings that we're able to detect with our method. Here, I'm showing a tryptophan DG formaldehyde crosslink, where again, we can monitor for the loss of DR. And when looking at the spectra, we can see that loss of DR, and we can also see tryptophan. And at first, however, I was a little concerned that I wasn't sure what that major fragment was, right? This 217 up here. But by drawing the analyte and taking advantage of the knowledge that I had gained by analyzing all of these other formaldehyde crosslinks, it didn't take long to figure out that that 217 is really just tryptophan with the formaldehyde linker still attached to it. And of course, for completeness, we did annotate the other recognizable ions in the spectrum as well, all indicating that this is a DG crosslink with tryptophan. And so when looking at the levels of these DNA protein crosslinks, here we notice that in the case of NNK only, there really was no prominent effect this time, if any at all. However, when we look at the co-exposure, we see that similar trend again, where increases are happening earlier, particularly the 5 week time point for these crosslinks. And so another thing that I also wanted to note here is that, as you may have seen, the error bars are quite high for many of these addicts. And so to us, this just suggests that we need to explore these trends further in similar samples and try and validate them in other models as well. So I also wanted to quickly show you some data for other addicts that we're also able to detect with our method. Here, I'm showing you the levels of various methylphosphate addicts. But unlike the previous classes of addicts that we've looked at, there doesn't appear to be any notable trends here. But for us, this actually kind of reinforced the idea that maybe we're actually onto something when it comes to the trends that we observed for these other addicts that we've looked at. And then again, when we look at abasic sites, that's another type of DNA damage that's actually a result, a downstream result of several types of DNA addicts. They also did not appear to show any clear trends either. And so before I continue, I just wanted to make a point that if anyone has questions about the mass spec methods that I've used for characterizing all these addicts, just shoot me an email. I'd be happy to chat. I'm always happy to talk about mass spec. So moving forward here. So we've characterized the profile of NNK-derived DNA addicts over time. However, to better characterize NNK-induced lung carcinogenesis, the next step would be to assess if any of these DNA addicts are inducing specific mutation patterns. So to do this, we used the same tissues from our animal model, extracted the DNA, and did library preparation using twin-strand biosciences duplex-seq mutagenesis assay kit in order to perform duplex sequencing. And so by doing this, we were actually able to create and sequence libraries for all 36 of our samples. If you have more questions about the number of reads per sample, the sequencing depth, etc., again, I'd be always happy to chat. And so first, I'd like to distill down this data into the overall mutation frequency. Here, you can see that the mutation frequency follows a similar behavior of that observed for the POBDT addict, right? Where there is an increase happening at the earlier time point for the NNK plus LPS compared to like the NNK only. As this increase in the NNK is taking longer to be observed, right? We don't see this increase in mutation burden for NNK only until the 15-week time point. And so seeing this data also confirmed that our model was working for generating this type of data as opposed to just working well for generating addict data. This really was the first time we had used DNA from one of our normal DNA extraction protocols to submit for duplex sequencing. And so breaking down this data further into the single base substitution mutation counts, you can see that there is an increase for these T to N mutations, right? So that's T to A, T to C, and T to G, right? These gray, green, and pink bars. And it's important to note that this increase for these T to N mutations is only observed in the 5 and 15-week time points, right? 5 and 15. And so I would say this suggests that our model is doing a good job at really capturing the pre-oncogenic process, right? Where we have a time point that is similar to the baseline mutagenesis, followed by 2 subsequent increases in mutation burden as the exposure duration progressed. And so next, when we break this data down into the trinucleotide context, we were able to see that there is actually a common pattern that was only observed in the samples exposed to N and K, right? We don't, for these T to N mutations, we don't see this kind of pattern at all really for the control nor the LPS only samples. And so next with this data, we used Dr. Ludmila Alexandrov of UC San Diego's tool. It's called a SIG profiler extractor to extract a de novo mutational signatures from this data. Essentially, this is an unbiased computational approach to identify patterns within the mutational data and create a signature that represents the different patterns that are observed within the data sets. And here we notice that one of the de novo signatures that was generated resembled the pattern that we had seen in the trinucleotide spectra for the NNK exposed samples. And additionally, also, there are some recently reported NNK and NNN mutational signatures that are very similar to our de novo NNK signature that we saw. And especially, right, when you look at the pattern that's observed for these T to N mutations, right, really it looks a lot like ours. And so this really suggests that we're moving in the right direction here. And so furthermore, using SIG profiler extractor, we were also able to generate something that's called strand bias data. And when you look at the strand bias data here, there's an increase on the genic untranscribed strand, right? It's these red bars are higher than the blue bars. And in this case, for these T to N mutations, this suggests that these mutations are a result of damage to thymine. And just to kind of help explain this a little bit, if this were the reverse with the blue bars higher than the red bars, then that would indicate that these mutations are a result of damage to adenine, because adenine is the complementary base of thymine up here, right? I know it's a little confusing. I'm happy to help explain this as well. It was definitely something I've had to work on understanding over the past year or so. But again, just as a reminder, our data is indicating that the mutations observed are a result of damage to thymine, right? And so, as you may remember, we were able to identify several thymine-related addicts that showed that pattern of earlier increases in the co-exposure. And so we're going to pay particular attention to these addicts and other modifications of T moving forward with this project that it is as it is still ongoing. And we're also going to try in the future validate these trends in other models as well. So, as a take home message, what I really want you all to understand is that we have new tools that can expand what we can see when it comes to both DNA adductomics and mutagenesis analysis, with the continued development of both high resolution mass spectrometers and genomics tools utilizing next gen sequencing, such as duplex sequencing approaches. And so what's especially exciting about the development of these types of approaches is that they can facilitate the integration of different omics approaches, allowing us to really dive into the molecular mechanisms and events that are at the basis of carcinogenesis by providing us the unique opportunity to try and understand what the molecular consequences of DNA addicts are, and vice versa, what molecular events are at the origin of certain mutational signatures. And so, all in all, by better understanding these mechanisms, we can set the stage for the development of biomarkers to be used for early detection and to inform cancer prevention, or even be used to help guide treatment as a predictor of prognosis. And so, with that, I have a lot of people to thank. I'd really like to thank the other members of my lab, especially my PI, Sylvia Balbo. I'd also really like to thank Pete Villalta. He really is an integral part to the success of my work and of our lab as a whole. And then I also really want to give a shout out to the Chateaubriand Fellowship, which is a fellowship provided by the French Embassy in the US, which they gave me the funds to go to France for 6 months and work at the International Agency for Research on Cancer, where I got to work under the supervision of Dr. Jerry Zavadil. And among other things, one of the main things he was doing there was he gave me a crash course on all things mutational signatures, which was really helpful to learn from a world expert in that field. And then also, I do want to thank the Renville County Walk in the Park. This is a county out in western Minnesota that has supported the work in our lab. And they provided the funds that we used to purchase that duplex sequencing twin strand kit, which really opened the door for us to start integrating our DNA addict data with mutation data. So we really appreciated that as well. And with that, I'll take any questions. Thank you very much.
Thank you so much for that presentation. And my laptop just forced me to get on my phone, so bear with me. But that was fantastic, and we have a huge audience for you today, which is really great. So, folks, please place your questions in the Q&A, or go ahead and unmute yourself if you have a question that you would like to voice. And while we're waiting, I'll go ahead and ask a question. You alluded to doing some validation in other models. And I was just
Mhm.
curious, what would that look like? What would be kind of that next step?
Yeah. So I think we will probably do is maybe do this like in mice again, look at the mice and see if we can see these trends again in another in vivo model. Because like typically when it like in our work, oftentimes it's nice to do things in vitro, then in
Hmm.
vivo, and then eventually try and assess this DNA damage in humans as well. And so what kind of the idea here is that we really want to like validate these trends in as many in vivo models as we can so that we can really kind of establish which DNA addicts may be predictive of eventual carcinogenesis, right? So we really just kind of establish these in vivo so that then in the future we can try and maybe monitor, maybe do like some biopsy analysis in humans as well or other models as well. So
Yeah, fantastic. That was helpful to hear. Great. And it looks like we have a question in the chat. Is that right? Um, Foster, do you want to go ahead and read it and then respond?
Yeah, sure. So, have you identified any new DNA addicts using your methodology? So that DNA RNA crosslink was a new DNA addict. So that was one that we had again we used our untargeted screening approach, and that's how we first detected that in mice. And then again we validated that in the rats as well. So we've seen it 2 times now. And with our untargeted screening approach, I mean, we're working on we've actually identified a couple putative addicts recently in various projects, and so we're really trying to understand. Try and figure out what those are. We kind of play with the mass spec spectra to kind of understand the building blocks of these potential DNA addicts. They're not always as simple as that DNA RNA crosslink, where like every product ion was something that we recognize and was clear that it's like a DA guanosine type of crosslink. So with these other potential addicts that we've identified, we're working on identifying them currently, trying to first is like see if we consistently see these addicts in multiple models, and if we see them in multiple models and we see that they're increasing from the after exposure, then those are the putative addicts that we really dive into and try and learn more about. So the answer to that is yes, and we're working on trying to solidify more as well.
Great. And I see Steve Hecht is unmuted. Go ahead.
Yeah, Foster, very nice presentation.
Thank you.
While you were talking about formaldehyde, it occurred to me that the formaldehyde could be generated downstream from inflammation that's caused by NNK. So how do you know that the formaldehyde adducts that you're seeing actually come from methyl hydroxylation of NNK rather than something endogenous?
Yeah, no, that's a great question, and that's something that we're still trying to evaluate as well. And so, I mean, when it comes to just something plain endogenous without exposure to NNK, I mean, we do see that for some of these crosslinks, they are higher in the exposed sample compared to the controls, but that increase is not always clear. So that's definitely something that we're working on, and that's why I want to try and validate these as many times as possible to see that upon NNK exposure, these formaldehyde crosslinks are increasing. And then also that's why we look at LPS, that agent that is inducing inflammation, to see because if they're increasing in the NNK exposed samples but not the LPS samples, then that leads us to believe that maybe they are due to the NNK exposure as opposed to the inflammation from LPS.
Yeah, you might try methyl-labeled NNK. That would be
Yeah, that's a great idea.
deuterium labeled. Right. One other question. So if I understood your mutational signature data correctly, you were seeing a lot of mutations at T.
Yes.
Is that due to the thymidine adducts of NNK? I mean, do you have any information on that?
That's what we're going to try and investigate moving forward. So now, just anytime we're working on a project related to NNK or tobacco-mediated carcinogenesis, we're going to really try and identify and try and target and quantify as many of these DT-related addicts as possible. And that's why we're really trying to develop more quantitative approaches for the various addicts that we're detecting. And that's why, when we do our untargeted adductomics to try and discover new addicts, we are definitely trying to pay more attention to thymine-related addicts that we're seeing. So yeah, just like in the future, we're going to just try and keep doing this where we expose something to NNK or another tobacco-related metabolite, and then or tobacco-related carcinogen, and just see if, again, we're seeing the idea is that we're going to hopefully see increases in these thymine-related addicts and also continue to see mutations or damage to T resulting in mutations. So we're just going to try and keep doing this over and over again to really see if this is a trend that we continue to see.
But the signatures that you see from the NNK treated rats, I guess these were, they're not so common in smokers, as far as I know.
Yeah,
Is that right?
that is correct. There are
So that
some.
means that NNK is not important.
I think that's what
Is that what
definitely
you like to say?
that's what some people would like to say for sure. So that's why we're really going. Well, the thing is, it's what's interesting is that this NNK derived signature that we're characterizing here, and that a few other groups have done as well. Like I know Shauna's done some work on it, and the group at IARC has as well. So the idea here is that pattern. If we can establish that pattern, especially so. If I go back a few slides just to kind of reiterate my point here. So it's yeah. So then like this pattern here that we see for these T to N mutations, the idea is that if we see this in NNK related samples and NNN and just over and over again for some of these projects that we're working on, if we can establish this, then what we can do, what's really cool with these tools that we use to extract these mutational signatures, these tools are also really good at diving into old cancer genomes and seeing that if any of these new signatures are found in any of these old samples. So they can we've sequenced tons of cancer genomes. And so then we can go back and look in those sequenced cancer genomes and see if this NNK signature is present in any of the smokers that have been sequenced in the past, if that makes sense.
Right. Thank you.
Yeah, thank you. Thank you. Great questions.
Great. Any other questions?
I have a question. Hello?
Hi, Eric.
Hi.
Hi. So for your studies, I know that you've explored the duration of the dose of NNK and the LPS. Have you looked at different levels of NNK dose? And do you see kind of like a similar dose response in your adduct levels?
Mm-hmm. I personally have not done that, but if I believe correctly, I mean Steve and Sylvia both have done that in the past. So personally, I have not. I don't want to speak on data that I'm not for sure about, but I believe that has been done.
Okay. Yeah, I'm just curious if the mutational signatures would, but for 15 weeks would be kind of similar at 5 weeks. If you
Mm-hmm.
had like a much higher
Yeah.
dose.
Yeah, it would be interesting to see how that changes with a change in dose. Yeah, and yeah, no, it's very curious because yeah, like as you've seen, like as the duration progresses, that those mutations do build up over time. But yeah, it is curious just if you hit it harder with a higher dose, do you see more mutations? So that's definitely something that we could look into.
Okay. Oh, thank you. And excellent talk. It was really well put together.
Thank you, I appreciate it.
Any final question for our speaker? All right, Foster. Well, thank you so much for joining us today and sharing about your work, which I understand is your dissertation or informing your dissertation. So,
Yes.
really exciting to see your training and the results so far. And we look forward to hopefully having you again when you're a little further along as well.
Yeah, for sure. Thank you. I'd love to come back.
Okay, great. Thanks, everyone. Have a wonderful Wednesday.
Thank you, everyone.